The overall goal of this scalable procedure is to isolate specific and high-affinity Fab-phage clones from a phage-displayed library using a high-throughput approach that enables parallel selection against hundreds of antigenic target domains. This is accomplished by first immobilizing purified antigenic domains in microwell plates. The next steps are to expose, capture, release, and amplify Fab-phage clones that specifically bind to microplate immobilized antigen in successive rounds of selection. During this process, the effectiveness of the selection is monitored using pooled methods at each step. The final step is to evaluate the specificity, affinity, and/or activity of the isolated Fab clones. Ultimately, specific high-affinity Fab fragments against large numbers of antigens are isolated in parallel, enabling the generation of antibodies useful in the analysis of entire classes of structurally or functionally related proteins.
The importance of this method lies in that it offers an alternative that can help to overcome the limitations and accelerate the pace of conventional antibody development technologies in either an academic or an industrial lab. The major advantage of this technique over conventional antibody development techniques such as hybridoma include that it doesn't require the use of animals. It can be used for virtually any protein, including toxic and non-immunogenic proteins. As we'll see, it's easily parallelized and ultimately yields renewable recombinant DNA encoding a high-affinity antibody specific for a particular antigen and functional in standard immunoassays in as little as 6 to 8 weeks. Given the importance of antibodies in research in biomedicine, we saw an opportunity to